All right, we are going to jump straight into Mendelian genetics with his laws and dihybrid crosses. So, just to recap, organisms can reproduce sexually or asexually. Sexual reproduction will require two parents, it produces genetic variation, and it uses gametes produced through meiosis. Now, asexual reproduction will only require one parent. There's no genetic variation, and this uses mitosis to create new offspring. So, cells in the body of sexually producing organisms can be either somatic cells, which are body cells, or gametes, which are sex cells. So, remember that your somatic cells will contain the diploid 2N chromosome number produced through mitosis. And there is 46 in humans, so you get 23 from mom and 23 from dad. And examples of your somatic cells are your muscle cells, your nerve cells, your blood cells, and your stomach cells. While gametes, they contain the haploid chromosome number, and they are produced through meiosis. So examples of this are going to be the sperm cells that are found in men and the egg cell that is found in women. Now, a chromosome is a thread-like structure made up of DNA. Chromosomes are found in the nucleus of each cell. A karyotype is used to show an individual's chromosomes. So the typical human karyotypes contain 22 pairs of autosomal chromosomes, or body cells, and one pair of sex chromosomes. So everything here in this row, this row, this row, and this row, up until this point, are all autosomal cells. Only down here in this corner, that is where you find your sex chromosomes. So you can tell by the karyotype that we're looking at, we have one X and one Y. So we know that we're looking at a male karyotype. So this karyotype shows the homologous chromosomes, which are the matching chromosomes from our mom and dad. So they contain the same genes in the same locations, but may contain different alleles or different versions of the same gene. So here's an example of one of our homologous chromosomes kind of blown up bigger for you to be able to see the bands, which is where your traits are stored. So a gene is a section of DNA that provides the instructions for making a protein. Since a gene is a piece of chromosome and we have two of every chromosome, we have two copies of instructions to make every protein in our bodies. Now, alleles are going to be different versions of the same gene. So we inherited a full set of chromosomes containing genes from each of our parents, but we may not have inherited the same version of every gene. So an example of this is mom could have given you a gene for blue eyes, and dad gave you a gene for brown eyes. Now, in this unit, we are focusing on heredity, which is the passing of traits from parents to offspring. Gregor Mendel, in his experiments with pea plants, he paved the way for what we know about inheritance today, and he provided us with three laws of inheritance: the law of dominance, the law of segregation, and the law of independent assortment. So the law of dominance is that a dominant allele will express itself over a recessive allele. So freckles is going to be big F, which are dominant over non-freckles, which is a little f. So if a person inherits the big F allele from one or both parents, they will have freckles. But if a person inherits the recessive little f allele from both parents, they will not have freckles. So remember that a genotype is the actual alleles inherited. So genes that code for freckles, such as big F, which is homozygous dominant, big F, which is heterozygous, or little f, which is homozygous recessive. Also, remember that a phenotype is the physical traits or characteristics seen in an organism. So, for example, this is the freckles that you would see on this person. The law of segregation is when chromosomes separate in meiosis and each gamete egg or sperm will receive only one chromosome from each pair. The chromosomes separate during meiosis. So if a man has alleles for brown eyes and blue eyes, he is heterozygous. So he has a big B and a little b for the eye color. So his sperm cells can contain the allele for brown eyes, big B, or the allele for blue eyes, little b. So each sperm only gets one of the alleles. Next, we have the law of independent assortment. The inheritance of one trait does not affect the inheritance of another. So each pair of homologous chromosomes consists of one chromosome inherited from the father and one from the mother. Each pair of chromosomes line up independently of one another in meiosis 1. Now, there are two different ways that chromosomes pair and can line up. And in humans, this creates about 8 million different combinations. But the law of independent assortment is completely random. Now let's start talking about dihybrid crosses. A dihybrid cross is used when finding the possible genotypes for offspring when you're considering two traits at the same time. So you are going to have 16 squares for a dihybrid cross. So let's start with example number 1. Cross two parent pea plants that are heterozygous for pea color and flower color. Note that yellow peas, capital Y, are dominant to green peas, which is little y. Purple flowers, capital P, are dominant to white flowers, which is little p. So we are crossing pea color and flower color. So let's write out our parents' genotypes. So parent 1 is going to be heterozygous for pea color and flower color, and so is the father. So both of them will share the same genotype of big Y, little y, and big P, little p. Right. So step 2. Write out the possible allele combinations for each parent that could contribute to the offspring. And we're going to place these on the outside of the dihybrid Punnett square. Remember, your first, outer, inner, last. We're using the FOIL method, like what you do in math class. So parent 1's combinations can be big Y, capital P, big Y, little p, little y, big P, and little y, little p. And dad's is going to be the same thing, since his genotype is exactly the same. Now, let's write out parent 1's alleles across the top x-axis of the square, and we're going to write parent 2's alleles down the side, which is the y-axis of the square. So I want you to keep in mind that you should place all the alleles for pea color first, and then pea shape. So place the dominant allele before the recessive allele for the same trait. So let's fill out our box here. So we got parent 1's genotype combinations, parent 2's genotypic combinations. All right, so let's start filling out our Punnett square. This is going to be just like our other Punnett squares. We're still bringing down and bringing over the information. Now we just have extra letters to work with. We're going to combine the alleles from the top and the left to fill in the square to determine the phenotypic ratio. Let's fill back out our box here. All right. So here's our box. Remember, anything on the top you're going to be bringing down. Anything from the left you're going to be bringing over to the side. So a yellow pea to purple flower is 1, 2, 3, 4, 5, 6, 7, 8, 9. All right. So we have 9 that are going to have a yellow pea and a purple flower. We know this because all of these that we marked off have a capital Y and a capital P. So next, let's look for a yellow pea and a white flower. 1, 2, 3, 3. So even though we have our capital Y's, we've got our little peas here, which means that it's going to have a white flower. Next, let's count our green pea with our purple flower. 1, 2, 3. All right, so we have 3 of those. Finally, let's count a green pea with a white flower. 1. That was pretty simple. So let's do another one. So tall plants, capital D, are dominant over dwarf plants, little d. And purple flowers, capital W, are dominant over white flowers, which is little w. So let's cross a homozygous dominant parent with a homozygous recessive parent. So step 1, write out the parents' genotypes. So parent 1 is homozygous dominant, which means that all of the letters are going to be capitalized because they are dominant. Parent 2 is homozygous recessive. So all of their letters for plant height and flower are going to be little letters. Now step 2, let's write out the possible combinations for each parent that could contribute to the offspring. And we're going to place these on the outside of the dihybrid Punnett square. So once again, we're going to do the FOIL method where we're going to go first to outer, inner, and last. So parent 1 can have DW, 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 and DW. So it's all going to be the same for parent 1. And parent 2 is going to be the same. Everything is going to be a little d and a little w for parent 2. So remember, parent 1 is going to go on the top of our box, and parent 2 is going to come down the side. Let's fill out our Punnett square here. Remember, we are using this to determine the phenotypic ratio of their offspring. So, remember, anything on the top is going to come down, and anything on the side is going to come this way. So let's start counting. How many tall plants with purple flowers will we have based off of this cross? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So we're going to have 16 tall plants and purple flowers. Let's see if we have any tall plants with white flowers. No, because everything is heterozygous. What about dwarf plants with purple flowers? Nope, we're not going to have any of those either. What about a dwarf plant with white flowers? Nope. Everything is heterozygous. All right, guys, I hope you enjoyed that. If you have any questions,